Hello, it's Dr. Williams here again. We're going to talk about B6 inheritance today. That forms part of your paper 2 for the AQA GCSE Combined Science Specification. That is if you're doing the trilogy. If you're doing triple science, you'll need to go and explore the extra content, but this is also on there as well. So let's get started and have a look at what this topic's about. So inheritance is really to do with all of the genetic material inside your cells, and the genetic material is found in the nucleus of a eukaryotic cell. It is stored as a molecule called DNA. You don't have to know it stands for deoxyribonucleic acid, just the letters DNA are useful. DNA is a polymer made of 2 strands, and it forms a structure called a double helix. So as you can see on that diagram, you've got those 2 twisted strands. They're connected together. If you're doing the triple content, you'll have to know how, but for now, we're just connecting together those 2 twisted strands, which form what is known as a double helix. And your DNA is organised into long molecules called chromosomes. And each individual chromosome is just made of a very, very long strand of DNA. And there are 23 pairs of chromosomes in human body cells. 22 of the pairs are numbered, and they're arranged in pairs because one comes from the mum and one comes from their dad. One pair of the chromosomes don't have a number, and that's because these are either X or Y chromosomes, and there are sex chromosomes. So if you're a boy, you inherit the XY, and if you're a girl, you inherit 2 X chromosomes. Different species have different numbers of chromosomes, but the same number of chromosomes is always the same within the species. So for example, humans have 23 pairs of chromosomes, and all humans should have 23 pairs of chromosomes, whereas another species might have a completely different number of chromosomes. And these chromosomes, they're made of DNA, and if you look at a small section of that DNA in a chromosome, you would find something called a gene. So a gene is just a small section of DNA on a chromosome. Each of the genes gives us a code for a specific protein, and that protein acts in our cells and as an organism as a whole to give us a specific feature or characteristic. So in that example, you can see there's the organism. If we look at one of the cells and take out one of the chromosomes and extract the DNA out and look at a small section of the DNA, that would be our gene. That gene would code to make a protein, and that protein would determine the colour of our eyes. So that would be our characteristic. The same genes, though, are on each pair of chromosomes, but there are different genes on the different chromosomes. If we just go back to this slide, what you'll see is that there are the same genes, for example, in the same position on chromosomes number 1, even though they are different chromosomes. And on chromosome number 2, we'll have entirely different genes to chromosome number 1. This relationship between genes and proteins is really important, and it's all living things have this relationship because the different genes have a specific code, the genetic code on the DNA sequence, which codes for an amino acid in a protein. So when you have a specific code, it means put this amino acid in this position. So what that gives us is if a genetic code changes, then the sequence of the amino acid changes, so you can make different proteins. And that's what gives us all of our variety and those different characteristics that we possess. One difference in the genetic code of a gene can result in a very different characteristic. So if you look at these examples, you can see the coloured beads represent the amino acid in that protein, and it folds into a very specific shape, which makes an enzyme. The same amino acids, but in a different arrangement and a different sequence, will fold into another very different shape, and that protein might form part of the cell structure. All of the genes and all of the genetic material inside a cell or an organism is called the genome. So not all of the DNA it codes for a gene. There are some bits of the DNA that don't code for genes. So we have this other term, genome, to represent all of that genetic material inside an organism. And the genomes can be sequenced. You can work out the sequence of their codes in order to be studied. And that's got great importance in science because it's useful if we're looking at what causes disease, how we can develop new drugs, see how humans have migrated in the past and track our migration patterns, as well as for studying evolution, not just in humans, but in many different species. And if we think about how inheritance really works and how this genetic material is inherited, then we need to think about reproduction. And there are actually two types of reproduction. The first we're going to look at is asexual reproduction, and asexual reproduction is very common in bacteria. It's also in other species as well. But asexual reproduction, the key thing is it involves only one parent. That means there's no fusion of gametes. And if you remember the definition of a gamete is a sperm or egg or pollen cell. This means if there is no fusion of gametes, that there is just a copying or cloning of that original parent cell. So there's no mixture of information. And the outcome is to have genetically identical offspring, identical to each other and identical to the original parent cell. The only type of cell division involved with asexual reproduction is mitosis. If we think about sexual reproduction, then, how organisms reproduce sexually, they require sex cells, such as sperm and egg cells in animals and pollen and egg cells in plants, and we call sex cells gametes. A fertilized egg cell, which we would call a zygote, contains the genetic information from the gametes of both parents, because the sperm and egg will fuse together to form the zygote. So what happens is the genetic material becomes shared in that zygote. Half of the chromosomes have come from the sperm, half have come from the egg. The zygote then divides by mitosis to form an embryo. And that means each cell in the embryo has the same genetic information as that original zygote. So more variety, there's more variation in your species. That gives you a better survival advantage. You're more likely to cope with any environmental pressures. However, you do need two parents, so it can be a slower process. But a lot of species reproduce sexually because there is more survival advantage. Asexual reproduction, on the other hand, there is less variation, so you have less of a survival advantage. However, you only need one parent, so that's easy to basically reproduce yourself when you need to. So it's a much quicker process. So you can see that there are advantages and disadvantages to species using these methods of reproduction. And some species have actually evolved so that they can reproduce both sexually and asexually. So, for example, some plants can produce the flowers, so they were able to produce pollen and eggs, and that pollen can be transferred to the egg of another plant. So that would be sexual reproduction. But they can also reproduce asexually and use other methods such as runners, and that's something that strawberries do. They send a runner out and then you get basically a clone growing from that runner. Or a potato, actually. If you take a potato from a potato plant and replant it, you would have a clone of its parent plant that you've taken it from. So you can see both of these strategies are useful for the potato and strawberry plants because it enables fast reproduction when they need to and a slower, more variety in their species if they need to use that strategy as well. So the thing about sexual reproduction is actually means that it leads us to another type of cell division called meiosis. And meiosis is the process of formation of gametes in organisms that wish to sexually reproduce. Meiosis is actually the dividing of a cell to produce gametes, and it is very different to mitosis. Mitosis happens for growth and repair. Meiosis is just to produce gametes. So it only happens in human males in the testes, where sperm, the gamete, is produced, and in females it will happen in the ovaries to produce eggs. During meiosis, the number of chromosomes actually is halved. So that means when fertilization does happen and the sperm joins with the egg, the full complement or the full number of chromosomes is restored. Because if you halve them to make the gametes, when they fertilize, they will restore the complete number. So these are the steps in meiosis. You don't have to know this in as much detail as mitosis, but it's still really important that you understand the key events. So the steps of meiosis are that, first of all, the DNA is replicated, which is the same as what happens in mitosis, but in step 2, the chromosomes line up in their pairs. So if you can remember back to mitosis, originally the chromosomes line up just one above the other, whereas in this case, they line up in their pairs. So pair number 1 and pair number 2 and so on. And then the first stage is to separate those pairs and then do a cell division. The remaining steps are the same as mitosis from this point onwards. So then the chromosomes line up in the middle of the new cell, and one set of each chromosome is pulled to the opposite ends of the cell. Then the cell membrane and cytoplasm divide. What happens is, instead of producing 2 daughter cells as you would in mitosis, you will produce 4 gametes. Each one has got half the number of chromosomes as the original cell, and they will all contain different genetic material because they will sort their chromosomes in a different way when they originally line them up in pairs. This means they've got variation. So actually the variation comes from the different gametes produced, and that variation can then be passed on to the offspring when they fuse with another gamete. Now this is quite a nice comparison between those 2 cell division processes, mitosis and meiosis. So it's just remembering and recalling that information from your paper 1 about mitosis, that mitosis is needed for growth and repair. It can happen everywhere in an organism. It can produce 2 genetically identical cells. It involves only 1 cell division, and the new cells have the same number of chromosomes as the original parent. Whereas meiosis is needed to produce gametes, only happens in reproductive organs, produces 4 genetically different cells. It involves 2 cell divisions, and the new cells have half the chromosome number as the original. If you remember that we said earlier that during meiosis the number of chromosomes is halved, so that when fertilization occurs the full number of chromosomes is restored, then this diagram really just clearly shows that happening. This means that when the nucleus of a sperm and egg fuse together during fertilization, the zygote actually inherits one of the chromosomes in each pair that they've now got from each parent. This means half of the genes they get will be from the female parent and the other half of the genes will be from the male. We can look at this in this sort of diagram. So if we're thinking about meiosis happening, they're the father's chromosomes lined up in their numbered pairs with X and Y at the end. Now they will randomly shuffle each of those pairs to make the gamete. So this is why each gamete has variation. If we look at the mother's chromosomes in their ovary when they're making an egg, they will again randomly shuffle each of those pairs into the gametes. So in this particular gamete, you can see that's how they've sorted them out. That already leads to variety. And when they meet, that sperm and that egg, you can see in that offspring 1, the pairs are matched up as so. If they have another child, offspring 2, they will have a different sperm made because the chromosomes have been sorted out differently during meiosis, and a different egg. So if you compare these 2, which are brother and sister, or actually they are brother and sister, you compare the 2. If you look at just chromosome number 1, then offspring 1 has got a blue chromosome from the dad and a purple one from the mum, and they've got a green one from the dad and a light purple one from the mum. So they have actually inherited different chromosomes, so they will have differences to each other. So each chromosome carries genes, and when we inherit chromosomes and their genes, we inherit 2 copies of every gene. Because all of the genes on chromosome 1, remember, are the same on the other chromosomes 1, but maybe with slight differences, but it's the same gene. So we get a copy of each gene from each one of our parents, but we actually inherit slightly different genes depending on the chromosome that we actually get. So this means that every offspring has a likeness to each other because they inherit some of the same genes from their parents, but they also have genetic variation because they inherit different genes to each other. If you know in this person, offspring 1 has inherited an X from their dad and an X from their mum, so they are female. Offspring 2 has inherited a Y from their dad and an X from their mum, so they are male. So you can really clearly see a characteristic difference between those 2 offspring. And we can work out the probability of which genes are inherited when using genetic cross diagrams, and we can also look at family trees to help us predict more about the genetics of families and inheritance as well. Now, these are both examples of a family tree and a genetic cross diagram. In a family tree, you'll see this A and B, they are connected together with a straight line. That means that they have reproduced together to produce these offspring. So the branch coming down from each one of them. So we've got a branch to D, a branch to E, and a branch to F. So they are all brothers and sisters, D, E, and F. And you see the line that connects F from G. So G is from another family. So G would be a female who's having reproduced with F. And you can see that they have had 4 offspring produced from them. So that's how a family tree works. A genetic cross diagram is a series of letters which shows you which genes are inherited. And we'll look at these in more detail in a minute. So this is an example of a genetic cross diagram to look at the inheritance of the X and Y chromosomes. So here's an example of a question you could be asked. A couple have 3 boys, they're planning to have another child. They think this child has a greater chance of being a girl this time. Explain why this is not the case using a genetic cross diagram. So what we have to remember is what we just learned is that the cells in the ovary divide by meiosis to produce egg cells. So they separate their pair of X chromosomes into different egg cells. That means there's 2 different types of egg cell produced with regards to the X chromosomes. They will have more than 2 different types overall produced because of all of the other shuffling of the chromosomes, but we are just focusing on the X chromosome. So in terms of that, there are 2 different types of X chromosome produced. The father, in the testes, they will produce 2 different types of sperm. They will randomly split up the X and the Y chromosomes and produce sperm with an X in or sperm with a Y in. Again, those other differences, the other 22 chromosomes are there, but it would be far too confusing if we started to look at all of those as well. So that's the first thing. That's where the principle of a genetic cross diagram comes from. The next thing is you can just use that diagram box to work out the chances, remember, of them not having a girl. They're not going to have more chance of having a girl this time. So first thing you do is you write the parental chromosomes in the genetic cross diagram. It doesn't really matter where you put the male and female, you just write them in those row titles and column titles. And then you imagine that this egg here is fertilized by this sperm, and this sperm is carrying an X. So the X will end up going into the egg, and the child produced will have 2 X chromosomes, one from the egg and one from the sperm. And you carry that on. So this egg and this sperm and the child produced will have 2 X's. And then this egg, along with the Y carrying sperm, and you will have XY and XY. So that is your genetic cross. Then you would indicate what the characteristic is that you would see. Now, we call a characteristic a phenotype. When we're looking at our genes, the resulting characteristic we can call the phenotype. So we see anything with an X, any offspring with an X would be female, 2 X's, and an X and a Y would be male. And you can see that's clearly shown by circling that and writing with an arrow female, circling and writing with an arrow male. And then we can sort of state or answer the question that it's asking you. So the percentage chance of each phenotype in this example is a 50% chance, okay? Or a 1 in 2 chance, or you can represent the 50% chance as 1-to-1 ratio or 0.5. You can't say 1-to-2 ratio because that isn't right. It's a 1-to-1 ratio. So there's a 50-50 chance that the offspring will be a boy and a 50-50 chance it will be a girl each time you have a child. So that's why it's not more likely to be a girl the 4th time you have a child if you've had 3 boys. And it's really important that you follow all of those steps. So before, the marks were given there, but let's just have a really good look. You actually get 1 mark for the correct parental chromosomes. Another mark, actually normally 2 marks for identifying the correct genotypes as a result. Another mark, and this is often the mark that's most missed by students, for circling and stating the phenotypes that you get. Always remember to do that bit. And then the final mark for basically answering the question is stating the percentage chance. Normally, if you get a genetic cross diagram question, it would be out of 5 marks. So try and aim to get all of those 5 key marks there. So the genes that you inherit from each parent might actually be different. The different versions of a gene are called alleles. So for example, you might inherit the gene for eye colour, which would say blue, make blue eyes. But from your mum, you might have inherited the gene for eye colour, which says brown. The eye colour allele blue and the eye colour allele brown will give different instructions to make slightly different proteins to give slightly different characteristics. So inherited alleles, one of those alleles needs to be dominant to the other allele. That means the phenotype you see will always be the phenotype of the most dominant allele. That's the allele that shows the phenotype no matter what other alleles are present. The other allele we would say was recessive, and you won't see this phenotype. So in this case, the blue eye colour allele is dominant and the brown eye colour allele would be recessive. And because this person has inherited the dominant blue eye colour allele, they will have blue eyes. It doesn't really matter what the other colour the other allele says to make, they would still have blue eyes because it is dominant. And we show dominant and recessive genes by representing them with letters. So the gene itself here is represented with an E for eye colour gene, and that capital E shows that blue eye allele is dominant. The recessive brown eye allele would be given the lowercase e. So there's some quite complicated but important keywords that I've used here. The first one is allele, which means a different version of a gene. I've not yet introduced the word genotype, but the genotype is basically a description of the alleles an organism has. So if we look at this first example on the right over here, this person's inherited the dominant blue eye colour allele from one parent and the dominant blue eye colour allele from the other parent. So they have the genotype capital E, two dominant alleles. The phenotype is the characteristic you see as a result of the genotype. So if you inherit two dominant alleles, you have blue eyes. Remember, just inheriting one of these gives you blue eyes. Because the blue eye colour allele is dominant, and a dominant allele means they are always expressed and seen in the phenotype, so even if only one copy of that allele is present. The recessive allele, on the other hand, if you inherit the recessive allele, as this in the middle has done here, so they've got the genotype big E, little e, that means they've got the blue eye allele, which is dominant, and the brown eye allele, which is recessive. You only see the recessive phenotype if you get two copies of that recessive allele present. So this person with one dominant, one recessive will have blue eyes, but this person at the bottom, who has actually inherited two brown eye recessive alleles, with the genotype little e, they would have brown eyes. Now, there's two other terms that we need to look at as well, which are called homozygous and heterozygous. So homozygous means that the inherited alleles are the same. So anytime you see homozygous, it could be a homozygous dominant, as in this case here, or homozygous recessive in the bottom example. They would mean the same alleles have been inherited. Whether they're dominant or recessive, it doesn't matter. It just means the same alleles. And heterozygous in the middle is when they are different. So hetero means different. So different genotype would have a dominant E allele and a recessive E allele. It's really important that you know these keywords and are confident with them, because you could be just given some of these keywords and have to interpret questions from them. So here's an example where these keywords are really important to understand in an exam question. So this incorporates a family tree, and it says figure 1 shows the inheritance of hair colour in one family, gives you a key, so the squares there are males. If it's a coloured square, it means red hair, not coloured square with brown hair, and a female with brown hair. Females are represented by circles. And it says brown hair is caused by a dominant allele B. Red hair is caused by the recessive allele B. And it asks, what combination of alleles does person 1 have? So this is a bit like a puzzle, really. You've just got to work it out from the information given. So what we can say is that 1 and 2 have reproduced, and they've got 3 offspring, 2 of which have brown hair and 1 have red hair. But 1 or 2, neither of them have red hair. So what that means actually we can already identify that that female with red hair must have 2 recessive alleles because they've got red hair. And we know that that's a recessive phenotype. We know that parent 1 would have a dominant brown hair allele because they've got brown hair. And parent 2 also has brown hair. Offspring 4 has got brown hair, so they've again got the brown hair allele. And offspring 5. But we don't yet know what this other allele might be for these 2 offspring or for the 2 parents. However, because person 3 has inherited 2 recessive alleles, it means that they have inherited one of them from their mum and one of them from their dad. So that means both persons 1 and 2 must have the little b recessive allele. It's because their parents have the brown hair phenotype, but the offspring has the recessive red hair phenotype, so must have inherited one of each of their 2 recessive alleles from each parent. So the person 1 must have the genotype big B, little b. They are heterozygous. And once you know that, they might ask a follow-up question like this one. It says, person 3 married a woman who was heterozygous with brown hair. So that gives you 2 bits of information there. And it asks you what is the probability of having a child with red hair. So because it says heterozygous, you straight away need to think, well, they're different alleles, but they've got brown hair, so it's got to be 1 big B and 1 little b. And then it asks you, what is the probability of having a child with red hair? So to calculate that, you need to do a genetic cross diagram. Now, before we showed the whole chromosome as X and Y for the sex chromosomes. In this case, now we would just show the alleles. So you show the dominant and recessive alleles. So this is person 3, and this is their heterozygous partner. And so what you would do is imagine that the sperm and the egg join together. It doesn't really matter, remember, which way around you put the parents, but it makes big B in that offspring, a big B and a little b in that one, and carry that through. And then you circle those different genotypes that give the phenotype of brown hair. So you can see you've circled those 3 and said brown hair, and circled that one and said red hair, and then answered what is the probability question, which is there is a 25% chance of having red hair. There are other ways of writing the 25% chance, for example, 0.25, but 25% chance I think makes most sense. And remember, for that 5 marks, 1 mark is for the parental genotypes, the parental alleles in the gametes. The other mark is for the offspring genotypes, and there are actually 2 marks available for that. The final mark is for stating the 25% chance of red hair, but actually the mark that everyone normally misses is for circling and identifying the phenotype. So make sure you don't forget to do that. So we also need to think about genetic cross diagrams with other things going on, not just inheriting characteristics, but thinking about things like genetic disorders. And genetic disorders are caused by gene mutations. And gene mutation is basically any change in DNA. And gene mutations can happen continuously all the time, and they're often repaired. And they can happen in our body cells. Sometimes, if they're not repaired, they can lead to become cancer. However, if mutations in DNA are inherited, if they happen in a gamete or are inherited via the gametes, they can affect the protein and therefore the phenotype of the offspring. And this is really how we get different alleles. They can also explain why and how we inherit genetic disorders. We need to bear in mind, though, is that most mutations have no effect on the phenotype. They just alter the gene slightly. It doesn't really affect the protein, and even if it did, it wouldn't affect the characteristic you see. Only very few mutations determine the phenotype, and then they will lead us to, over time, introducing different alleles into a population. But sometimes the mutations can be detrimental to a species. So this is the case for some of them, which we call genetic disorders. Polydactyly is an example of a genetic disorder. It's a dominant inherited genetic disorder and causes a person to have extra fingers or toes. And it's caused by a mutation in the genes which affect the patterning of the limb development in the embryo. But it only really affects extra fingers or toes, and people can have them surgically removed if they want, and it is not a life-threatening disorder, but it is dominant, and that's what you need to know about it. An exam question you could be given is one like this, where it says polydactyly is a dominant inherited disorder. If big P represents the dominant allele and little p the recessive allele, what are the chances of having children with polydactyly if one of the parents is heterozygous for the disorder and one is homozygous without the disorder? So what you need to do from the information in the question is, look, there's heterozygous. That means they are big P, little p for their genotype. Homozygous means that they are two little p's. So again, if it's saying what are the chances of having a child with polydactyly, so you do a genetic cross diagram, you show those two genotypes, and you circle the different genotypes produced, identify if they do or don't have polydactyly, and state a mark for what are the chances. So there's your 5 marks answer to that question. Another genetic disorder is cystic fibrosis. Cystic fibrosis is one of the most common life-threatening inherited diseases in the UK. It's caused by a recessive allele and results in disorder of cell membranes. This mutated allele will affect the movement of salt and water. This means there's a big, thick mucus that builds up in the lungs and the small intestine and can lead to difficulty breathing. You have to have physiotherapy and take drugs in order to make that mucus become less sticky and get rid of it out of the lungs. If you don't do that, there's a chance that you will get symptoms that get worse over time, that you will start to get damage to the lungs because of regular infections. So it's really important that people with cystic fibrosis do manage the condition really well. You can be a carrier of cystic fibrosis. That means that you're heterozygous. So in other words, you have the dominant, or what we would say would be healthy allele, and as well as the cystic fibrosis recessive allele, which causes the problems with the cell membranes. But because you're a carrier, you've got the heterozygous genotype, but because the healthy allele is dominant, you don't have any characteristics of disease, which is why you would be a carrier for cystic fibrosis without symptoms, but carrying the recessive cystic fibrosis allele. So here's an exam question which challenges that knowledge. So it says around 1 in 25 people are carriers for cystic fibrosis. Cystic fibrosis is a recessive genetic disorder. You are meant to know that it is recessive. You might be asked if it is recessive or dominant. It says if F, capital F, represents the dominant allele and little f the recessive allele, what are the chances of having children with cystic fibrosis if both parents are carriers? And so remember, carriers means that you're heterozygous, but you don't have any characteristics of the disease. So the first thing to recognise is that. So that means that you're going to be heterozygous and have a big F and a little f as a genotype. And it says if both parents are carriers, when you've done that and you've written the parental genotypes into your genetic cross diagram, when you've written all of those offspring genotypes in and got 2 marks for that, and circled the genotypes which give each phenotype. So the big F, if that's present, remember they do not have cystic fibrosis. Little f's do have cystic fibrosis. You can work out there's a 25% chance. Just be very careful. I know we've had a few questions that say 25% chance. It's not always a 25% chance for your question. You've really got to look at the question and work out your genetic cross diagram for each example. Also, with this mark scheme, it's very good, really, that it's broken down in this way. If you do make a mistake with one of the parents, for example, if you put big F as a mistake, and you do everything else correctly, then you would still get most of the marks. Thing about genetic disorders like cystic fibrosis is it is a life-threatening inherited disorder, and there are others which are also life-threatening, and these we want to really know if you have inherited them or not. So the advances in gene technologies means you can carry out screening and testing to check if you've got a genetic disorder or a chance of passing on a genetic disorder to your offspring. And you might be offered this genetic test because your doctor thinks you might have a health condition caused by a genetic disorder. Someone in your family might have a health condition which you know is caused by their genes. Some of your close relatives even have a particular type of cancer in particular that could be inherited. You or your partner have a health condition that could be passed on to your children, and you want to check that you're not going to do that. or it's part of a routine screening program. That means something that they are checking as a genetic test to check the general population. So there are different ways you can carry out genetic tests. You can get a swab of cells or blood samples from an adult any time after birth. And currently, cystic fibrosis is screened after a child is born in the UK. So what they do is they prick the heel and they take a sample of blood. You can also do genetic tests using ultrasound scans if you're pregnant and you think your child might have a genetic disorder that you can see, for example, something like polydactyly. That can be picked up during an ultrasound scan. Genetic tests, though, can also be performed on embryos, and that is during the IVF procedure. So if you remember the IVF procedure from B5, you would take the egg samples out of the female and add sperm sample to the egg so it fertilizes in a dish. Then you allow those fertilized eggs to grow into embryos. What results is a number of different embryos, and you choose one to be implanted. Before you make that choice of which embryo you're going to implant, you could actually test all of the embryos for particular genetic disorders. So that forms part of that IVF procedure. However, if you're already pregnant, you didn't have IVF, and you didn't know if you kept passing on a genetic disorder, you could test the fetus itself with an amniocentesis test or chorionic villus sampling. Now, you don't have to know the names of those tests. This is just fetal testing during pregnancy. But you can see they're both carried out at relatively early stages in pregnancy. Now, there's many reasons for having a genetic test, but there are some ethical considerations to think along the way. So you have to decide if the embryo does have a disorder and you've done a test while pregnant, whether you're going to terminate the pregnancy, which is have an abortion. Obviously, that's a very difficult decision for families to make. And does have an abortion imply that people with certain genetic problems are undesirable? This actually could really increase discrimination. If a child is born with a very serious genetic disorder, they may need lots of hospital treatment or medicine to support them. That's going to be costly for society as well as the parents themselves, and some parents might not be in a position to support that. If you're going through the IVF process and testing embryos, that is an expensive process. And also, if you're going to do amniocentesis or chorionic villus sampling, so testing the fetus there's a small risk of miscarriage, so you could actually lose the baby. And there's also a health risk to the mother. You might introduce virus or infection by just having the test. So there's lots of considerations around genetic testing. If you do have a child with a genetic disorder, then there is currently no cure for genetic disorders because it's in every gene, in every cell. All you can do is manage the symptoms. In future, we are hoping to be able to repair genes using something called gene therapy. And gene therapy is an experimental technique, but it allows healthy versions of genes to be inserted into patients for that gene to be expressed in the cells of the patient. If you can get a healthy version into the copies of all of the cells which need it, then what would happen is you would not see any effects of that disorder anymore. So, for example, if you could put the healthy gene into cystic fibrosis patients, then there would no longer be problems with the membrane and transport. And there's a diagram of how gene therapy would work. You would cut out what's called the normal or healthy allele, make lots of copies, and transfer that into the cells of a person with a genetic disorder in order to make that healthy gene work inside those people. So some of these ideas start to link into the general idea of how we alter genes in different species. And the differences in genes in different species, as we talked about earlier, is called variation. And we get more variation when we sexually reproduce and less variation when we asexually reproduce. And variation is caused by, as you've learned, the different genes or alleles that you inherit, but it can also be caused by the environmental influence we have, and both are genes and environment. So variation like hair colour, your hair might go lighter in the sunshine, but actually the gene for hair colour is inherited. And it was Charles Darwin that came up with the idea of evolution. And evolution is the change in these inherited characteristics of a population of a species through a process called natural selection. And that may over time result in the formation of a completely new species. And this theory of evolution by natural selection was first described by Charles Darwin in his book, which he called On the Origin of Species. And just remember, a definition of a species is a group of organisms which are able to breed together to produce fertile offspring. And his idea about evolution gave rise to the idea that all species of living things have evolved from very simple life forms more than 3 billion years ago. You do have to know those dates and those times and the actual figures for this. And at first, this idea that we all evolved from simple life forms that over 3 billion years ago was quite a revolutionary idea, and it was only gradually accepted over a longer period of time, because this idea that Charles Darwin had challenged the theory that God made all the animals and plants that live on Earth. There wasn't a lot of evidence at the time that was to convince the scientists that were around. And the mechanism of genes and the word gene wasn't really known. We didn't really understand inheritance until about 50 years after The Origin of Species were published. So it really was quite a novel way of thinking about how genetic information was transferred, considering we didn't know about genes. And Darwin's idea of natural selection was basically could be summarised by this little diagram shown on this side. So this is the mechanism of natural selection which drives evolution. So natural selection really relies on that variation, and variation within a species arises because of mutation in DNA. What that variation will result in is different phenotypes which are suited to the environment. So if they're best suited to the environment, that will help a species, an individual, to survive. And if that individual survives, they're more likely to breed. If they're more likely to breed, they will have many more offspring with that particular allele with the mutation, with the suitable phenotype, which again allows them to survive and breed. And we go round in a cycle of survival and breeding and passing on those really useful alleles to the offspring. And if this cycle continues, we get a species evolving over time to make them more suited to their environment, but still being able to adapt if there's still a lot of variety through sexual reproduction. This is a really nice, neat summary if you're ever asked to describe evolution by natural selection. So you always start with the variation in the phenotype shown by the organism and say it's due to mutation, how that variation in the phenotype allows individuals to survive. So there's more survival of individuals with that particular mutation, and breed, and then have more offspring with those alleles which cause the variation. Okay, so if you remember that cycle and just sketch it out, if you're asked a question about natural solution, that's a really good exam technique. Talking of exam technique, here's a practice question. It says the vole is a small mouse-like animal. Voles are found on some cold islands in the north of Scotland are much larger than voles found in warmer areas such as southern France. Explain how natural selection may have caused the northern voles to be larger in size. So using that idea of that cycle, what you would say is there is genetic variation caused by mutation in DNA, which causes the larger vole phenotype. You'd say how this helps. So the larger voles have more fat to insulate them. So the voles with more fat are more likely to survive in cold temperatures because they are better insulated. These voles go on to breed and produce more offspring, which have the alleles for the larger vole phenotype. Over time, and lots of cycles, and if there are environmental pressures, you might get a new species evolving. So if two populations of one species, it's more likely to happen if those new species are if that population is separated by some sort of barrier, and you're more likely to have different species formed if they can no longer breed together. Species can be shown on evolutionary or phylogenetic trees, and this is a phylogenetic tree diagram here. And what you'll see is that at each branch of the tree, you get a different species. So the current species, A, B, C, D, E, F, and G, they're present day. And as you go down towards the sort of the trunk of the tree, I suppose, that's more time longer ago. So these present day species, you can see at each branch where we get a branch coming away from each side, you will get that would be a common species, an ancestral species, where that population has basically split apart and we've now got different species evolving. This common ancestor species here is completely different to any of the A, B, C, D, E, F, or G species which exist now. And phylogenetic trees are really useful to looking at those relationships. And each of these branches where you see, that's where their common ancestor will have diverged. And we can use lots of classification data for living organisms and fossil data to piece together how all of these different species are related. And you can see the links of an evolutionary tree will show these shared or common ancestors really clearly, and the current species are always shown sort of at the ends of the branches. And much of the evidence for these phylogenetic trees comes from fossil evidence. And this fossil evidence really supported Darwin's idea of natural selection as well. We also have evidence where we can see evolution in action, where it's a development of antibiotic resistance in bacteria. This again relates back to paper 1 content. So if we go through that, we can see there's bacteria which have a mutation in the DNA which causes them to be resistant to a certain antibiotic. When they're given the antibiotics, most of the bacteria die, but the ones that have the mutation survive. Those resistant bacteria with the mutation become more popular and more common because they're able to survive and reproduce. And eventually, you will get more bacteria, and they're all resistant to the antibiotic because they all inherit this allele. And this really does support Darwin's idea of natural selection. It's been shown that this characteristic has been passed on from one bacteria to another in their genes. So all this evidence really helps to underpin the ideas about evolution. Fossils are a really good, useful piece of information. They can show us the remains of organisms from millions of years ago, and they are found in rocks. So a fossil is just the remains of an organism from millions of years ago. And the fossils are mainly formed from the parts of the organisms that don't decay because one or more of the conditions needed for decay are absent. And that comes into thinking of the WOW to help us remember that. The conditions for decay are water, oxygen, and warmth. So the word WOW can help us to remember what is needed. So if any of those are missing, you're much more likely to form a fossil after death. When parts of the organism are replaced by minerals as they decay, but that happens at different rates, there's a greater chance of forming a fossil with that. And you can also get fossils from preserved traces of organisms. So they might be footprints, burrows, and rootlet traces, and they all help to show us what organisms were like a long time ago. And they can provide really useful evidence for evolution. However, we don't expect every species ever to existed to have formed a fossil. We need the right conditions. So there are many gaps in the fossil record. As you can see, there's the fossil record of horses. So they're the fossils that we've got of what horses and their hooves look like from modern day to a long time ago. And there's obviously gaps along the journey of common ancestors that would have helped to explain how the horse's hoof evolved. And remember, life on Earth is thought to evolve over 3 billion years ago. So if there's any life on Earth from 3 billion years ago, it's really difficult to find because we think that was bacteria. So there were no hard parts, and bacteria wouldn't leave many remains. Also, 3 billion years ago, the Earth has gone through a lot of geological change in that time. So volcanoes will have altered the rocks over time and therefore destroyed any fossils that might have been there. We also have evidence for evolution in the idea of extinction. So we have fossils of dinosaurs, which we know are no longer able to survive. So many dinosaurs were unable to evolve by natural selection for their species to survive to present day. The dinosaurs actually became extinct 65 million years ago, and extinction means when there's no more individuals of a species remaining. So when species become extinct, like the dinosaurs, and there's many of them, we call that a mass extinction event. An extinction is when a species is no longer able to reproduce and survive. And so variation within a species can really help natural selection to happen and evolution to happen. And if there's more variation, it's likely there's more chance that your species will survive and less chance it will become extinct. Extinction can become more likely if there are these factors present. Number one, changes to the environment over geological time. That can obviously put pressure on a species. If there's more successful competitors, that can put pressure on a species. New diseases or new predators. Just through the cyclical nature of speciation, those changes over time can actually lead to extinction rather than survival. Or you could have a single catastrophic event, for example, a massive volcanic eruption or collision with an asteroid might cause extinction events. To remember those different ways or impacts that these factors can have on extinction, you can think of this as extinction can destroy popular species carelessly. So the E stands for environment, C for competitors, D for diseases, P for predators, S for speciation, and C for catastrophic event. We can obviously put things in place to prevent extinction. Humans have a great impact on this. Governments and individuals can create and fund protected habitats, put laws in place to make hunting illegal, make selling products from endangered species illegal, fishing quotas or net sizes if you're looking at saving fish species. You can work with other countries to protect species. You can try and achieve net zero carbon to reduce the effects of climate change, and reduce, reuse and cycle to avoid pollution and help support different species in their habitats. But not all organisms change through natural selection. Sometimes changes happen because of human intervention. And when we change characteristics, it's called selective breeding. And selective breeding is basically a type of artificial selection where humans breed particular plants and animals to select for certain characteristics. This can involve choosing the parents with the characteristic you want of the same species. These two parents are bred together, and the offspring are selected, which also have the desired characteristics, and then they are bred together as well. And this keeps continuing until the offspring shows the desired characteristics. So this can take quite a long while to do, and you need to keep breeding the organisms together and looking for the desired genes in your offspring each time. So there are some advantages to doing this. The first advantage is you're going to select for things that you want, so you're going to have greater yields. You can breed more domesticated animals, so they are, easier to look after. There are financial gains, and people don't mind this as a way of altering genes. Disadvantages are that there's less genetic variety. There are more risk from diseases. There can be unforeseen issues, so you might breed characteristics you don't want, like aggression or sterility. And sometimes it can be so selective bred that it becomes cruel to the animals. So we can breed chickens so they're so big they're unable to walk. So when you're thinking of the advantages, FAY might be useful to help you remember. So finance, acceptable and yield. And good could help you to remember genetics, unknown, risks and disease. So another way of altering a species genome is by the process of genetic engineering. And genetic engineering involves modifying the genome of an organism by introducing a gene from another organism to give a desired characteristic. So in genetic engineering, genes can be cut out of chromosomes of humans and other organisms and can be cut out and transferred to cells of other organisms. So you can basically transfer any gene from any organism into any other organism. So genes can be transferred between the same species or the different species. So some examples of genetically modified organisms are here. So you can see this tomato on the right has been genetically engineered. It's called the flavor saver tomato. It has actually got a protein in it from an Arctic fish. And that protein helps the tomato to be more resistant to colder temperatures. Golden rice, another example here, has got extra vitamin A. So it's got the vitamin A gene inserted into it, whereas normal rice doesn't have it. And this venomous cabbage has a toxin from a scorpion in the cabbage, which isn't toxic to humans, but is toxic to caterpillars. So a key advantage of that is you would use less pesticides in order to kill the caterpillars on the cabbage. So that could have a really good environmental impact because it would then result in less pesticides being used and less other organisms being killed, like bees or any other sort of insects that might be around those cabbages. If you're doing the higher tier paper, you do need to know the process of genetic engineering and the main steps involved. So the first step is that you would use enzymes to cut out the required gene, the one that you want to transfer. This gene is then transferred or inserted into what we call a vector. And a vector is a plasmid or a virus that is going to carry your gene into the organism that you want to genetically modify. This vector inserts the gene into the required cells during an early stage of development. So normally at the embryonic stage, if you're looking at animals, or, it's very easy to do in bacteria at any point. These cells then grow by mitosis, and because they're grown by mitosis, they're copying all of the genetic information into the new cells they produce, including the one that has been transferred. So that means that all of the cells in that organism have their own DNA plus the DNA that you have genetically inserted. Now, a good example of this is shown here in this diagram, where you've got this human cell with its chromosomes, and they've actually cut out the insulin gene and put that insulin gene into the plasmid of a bacteria and inserted that plasmid into a bacteria. When that bacteria grows and reproduces itself by asexual reproduction, all of the resulting bacteria will actually have the insulin gene in them in that plasmid. So you can get that insulin gene expressed, get the protein out, and you extract it to be able to give it to people who are diabetic to be able to use human insulin genes to treat the disease. Some advantages and disadvantages of genetic modification are that the genetic modification is generally a really good thing for increasing yield or popularity of things and making them more popular. They can be used to help treat illnesses in a large-scale way, including some inherited disorders, as well as things like producing insulin for diabetics. You can also grow resistant crops to herbicides, as we've seen, or insecticides, so you don't have to use those harmful pesticides. You can even now genetically modify organisms to be resistant to climate change, so that actually they can be more resilient in drought conditions or in really like flooding environments. So there's lots of advantages to using genetic modification, but there's also disadvantages. One of the main concerns is if you insert a new gene into a crop, how do you know where that gene ends up if like a pollinator comes along and transfers it into a plant somewhere else? So you can't really track where the gene goes. Also, we're not really sure of the long-term or short-term effects on human health. For example, if a protein from a fish is inserted into a tomato and people are allergic to fish, how might that affect those people? So some of those health issues haven't been fully explored. Another disadvantage is seen here with the genetic engineered glowfish. They were engineered to monitor pollution in rivers and released into the wild, but they were soon predated upon because they were really obvious to predators. They're now used as a novelty fish, but sometimes the unexpected outcomes from that genetic engineering can have things that we don't expect to happen. So that's also something to bear in mind. All living things, though, can be actually classified and worked on and looked at together. But if we are communicating about living things with other scientists around the world, we sort of need a system to be able to call them the right thing. So Carl Linnaeus developed this system of classification to be able to group these organisms and look at them and communicate about them in a really effective way. And Linnaeus developed a system a long time ago in the 1600s where they grouped organisms into kingdom, phylum, class, order, family, genus, and species. I'll talk to you about how he did that later on. These organisms, when you get to the end, the species name, are then given a Latin name, and we call this the binomial system. Bi meaning two, nomial name. And this is a two-part name which we can understand. Scientists all over the world can all understand it. The first part of the organism name is the genus, that classification group. The second part is the actual species name. And species names for every single living organism are always given in italics and in Latin. This genus has a capital letter and the species starts with a small letter. And this really rigorous and clear naming and classification system allows scientists all over the world to communicate about species without confusion. So, for example, here is the polar bear. Its name is Ursus maritimus, and you can see that Ursus is the genus part, the maritimus is the species part, and every scientist around the world working on polar bears will know that as that species name. However, things changed since Linnaeus' time in the 1600s. We started to identify cells, we started to see things with microscopes within the cell, and we understood more about genetics. So nowadays, we classify things in a slightly different way because of these developments. We can actually use genes and proteins to help us understand the relationships between all the different species as well. So you can see here, this is hemoglobin from a human, a horse, a sperm whale, and a sea turtle and tuna. We can start to look at the similarities between these specific proteins in all these different species and work out their evolutionary relationships, not just looking at things like their bone structure and things like that in the way that we would have done before we understood about genes and proteins. So the advances with technology mean that we can classify organisms a lot more efficiently. So we needed an adaptation to the Linnaean model, and we did that through the scientist called Carl Vose, and he developed a 3-domain system. So what he basically said was Linnaeus model works fine, but before we get into our kingdom, we need to think about these domains. And the domains were that archaea, which are primitive bacteria, which live in extreme environments and have been on Earth for billions of years. Bacteria, which we call prokaryotes now, we'd say true bacteria. And eukaryotes, which includes all protists, fungi, animals, and plants. So that's eukaryote or eukaryota. And this domain system fed into the Linnaean system of classification. So overall, we now have this system of classification. The domain at the top, which is the main group that you're in. The kingdom is the group within that. And then phylum, group below, class, order, family, until we get to the genus and species. And remember, those two give us our species name. So here we can see is the fox. The domain of the fox is its eukaryote. The group of the eukaryotes it's within is in Animalia. It's in the phylum Chordata, the class Mammalia, the order Carnivora, the family Canidae, the genus Vulpes. So the species name is actually vulpes. So the red fox, as we would know it as, is actually got a Latin name of vulpes as its binomial name. And a way to remember this classification system is to remember this little rhyme of do keep pond clean or froggy gets sick. So you won't have to list the whole of the domain, kingdom, phylum, class. You might be given this with some missing gaps that you have to fill in what's missing. So this little rhyme can help us to remember the order of these words. Okay, that's it now for B6. We've actually finished the whole of this topic. It's rather a big one. I wish you the best of luck for any exams that you might be having to do, and I hope this has helped to understand some of that content in a little bit more detail. Good luck, and hopefully my tips will help you.